Ćao i dobrodošli u još jednu epizodu Dive In podcasta. Nastavljamo dalje sa naukom i danas imamo jednog posebnog gosta iz Amerike koji nam dolazi sa Harvard univerziteta. I on je član grupe profesorice Dragane Rogulje koju svi znate iz naše emisije o tome zašto i kako spavamo. Ethan Glantz je student na doktorskim studijama na Harvard Medical Schoolu i on izučava kogniciju, pamćenje, učenje i taj deo aspekta u korelaciji sa spavanjem. Za potrebe ove epizode mi ćemo morati da se prebacimo na engleski, ali ćete imati titlove. Ethan, welcome and it's such a pleasure having you here.
Yeah, thank you for having me.
Welcome to Dive In podcast where your professor was. So it's really nice to have you here. And then I was looking forward to this American perspective of everything. Even the education systems, not just the fact that you're a PhD researcher at Harvard Medical School, but just like your own path of getting there. I think that would be really interesting for our viewers to tell them more about that. So Ethan, where do you come from? Who is Ethan Glantz?
So, yeah, I'll try to answer that. I'm from New Jersey.
Okay. We love New Jersey in Serbia.
Really?
Yeah, because we had the Jersey movie, you know.
Jersey Boys?
Yeah, no, the one when Ben Affleck has a kid. You remember that one? Yes,
I don't even know it.
It's a Jersey girl. Okay.
Okay. Yeah, it doesn't have as high of a reputation in America for some reason. I liked it. So yeah, from New Jersey. I grew up and for like since I was 5, wanted to just be a doctor and a physician. Because
and it's a thing in America. We talked about that.
It's a big thing in America. There's also like, when you're 5 years old, there's like 4 jobs. There's like doctor, firefighter, doctor, lawyer, and policeman, and like astronaut. Those are the only jobs. And so if you are someone who's like a little bit more interested in science, it becomes this kind of cyclical thing where, oh, that must mean be a doctor. And a lot of people don't really exactly understand what the job of like a scientist or a researcher is. And so my parents, you know, bless their souls, they didn't really know. They were like, oh, you want to be a doctor? And I was like, yes, I do. So for the age of 5 to the age of 18, when I left home, I did everything I could. I was the president of the Future Physicians and Nurses Club. I was an EMT. I shadowed doctors. I saw a couple surgeries. I really took a lot of steps. And then when I got to college, I went to a small school called Brandeis University outside of Boston. And I just realized that was not at all the thing I was interested in. I don't like people enough, I think is critical. And it's a hard life, and it's not, you know, I didn't want to practice medicine. I realized I was interested in the more creative process that is associated with research. So,
Thanks God for the undecided year in America, then you can, you know, or the first general year.
Yeah.
Because we don't have that in Serbia. Like if you chose law is going to be...
Yeah, just
or if you shift, then go from the beginning. It's not going to be...
If I was in, you know, especially like in a lot of places in Europe, like in Germany, for example, you want to go to medical school when you're 18 years old, you go to, I just would have been in medical school. Or maybe I wouldn't have gotten in. My grades were like kind of so-so. But I, thank God, I went to, you know, in colleges, you could also apply to some universities, you have to apply to a specific college at the university, in which it is you have to be an engineer, you have to be this. But at a place like where I went, which is a liberal arts degree, which is you pick a major when you're like a second-year student. And so you have some time to think about it and take the kind of general courses you need to. And so in those first 2 years of college, I took some classes, you know, basic science classes. But I totally didn't want to do medicine anymore. And I had like this weird obsession. It was a bad phase of my life where I was like, went the other direction and like thought I was going to be some comedian. Yeah, it was terrible. Thank God. Don't ask me to do any jokes because they weren't good. But then I had an existential crisis, dropped out of some classes. I got 6 one semester, realized I don't think it was really what I wanted to do. And then just kind of got lucky. Brandeis University had some research on its campus. It's a small school, but there's a lot of good research there. And one of my friends was doing some research, and I never thought I'd be into it. All of my lab classes, I like hated. I would like break things. I like did terrible. Thought it was the most boring thing in the world. But then I realized it was an interesting thing, and that I could have fun doing it. So I like slowly like kind of crept into there, but it took 20 years to figure that out. Yeah.
And then you got into neuroscience.
And I got into neuroscience. So I was.
Did you got to love all those classes or no?
I have never liked a single class in my life to be totally.
Thank you for sharing this. I think it's
totally
going to be a huge comfort,
Yeah.
not to Serbian students, but to every student who's listening to this and struggling and having existential questions and, you know, asking themselves like, is this even right for me? I hate it. I just hate it.
You know, it's like there are some things you can get out. I'm very cynical, but there are some things you can get out of a classroom, especially if it's like your first time taking like a big intro class. You can learn some concepts and some ideas about something. You're like, oh, I didn't even know what I could look up before to know what there is to learn. So barring those like 1 or 2 times that's happened in my life, I really found classes to be pretty prohibitive towards my own education. And, you know, if I didn't grow up in the age of the internet, I truly do not know how I would have learned anything. Yeah.
Did you take some extra classes or credits or something extra from the university? Like,
nope, I did the bare minimum to get. I
took survive.
I took extra classes, but that was like me. Like my first semester, I was like, oh, I'm gonna take a music class, and like I don't know what the hell I was doing. Like I was like almost did some theater stuff. I like almost did a bunch of things, and then I just like needed to leave. I took more classes than I needed to, but none of them that were really truly helpful for where I am now.
From this point, we're talking you're at Harvard University, and then we are taught here that only the best geniuses or the people with loads of money could get into some school like that. And then you just told us that you had bad grades. So it doesn't like what happened.
Yes, no, this is important. You know, I'm trying to walk like a fine line here in how I describe this, but my grades were like fine. They weren't that bad. I did fail.
They were like perfect in terms of 4.0 GPA.
I failed 2 classes, one of which was not for my major. It was not a science class. And my other one was a computational neuroscience. I failed. I got like 3 or 4 C's or C minuses. And my grades were just like fine. My final semester GPA was only a 1.8, which out of 4.0 is really bad. So what ended up happening is I was a little bit too much on the side of I knew, because there's a truth here, that grades are really, really, really for research programs, not necessarily what they're looking for. It helps quite a bit. And certainly you'll find a lot of places that have where students do pretty well and have high 3.8s or something, mostly A's. But I just knew that, you know, if I wasn't failing every course, and if I can do enough research, that is the thing that ultimately is what weighs the most for an application. And so what I did, so I got into research. Even my research experience was not very good my first couple years in undergraduate. I was part-time student, part-time researcher, which is hard, was for me to do well at both. I was in a lab where I didn't fully understand the type of material that we were studying. I knew things. I'm underselling myself a little bit here. But usually when you're an undergraduate in a lab, you do a thesis, a senior thesis. And that is like the culmination of your work. And normally that's like enough to get you right into PhD programs if you do it well enough. But my thesis was so bad that my thesis defense, the big thing that's supposed to be a little bit ceremonial and a little bit not serious, was actually canceled like 3 hours beforehand. It wasn't good. I don't think it was fair that they canceled it. They should have just let me do it and told me later that I wasn't going to get honors. But it was a total like debacle. I invited all my friends, my family. They drove from New Jersey, and I was
presents, excitement.
And so totally not cool and fun.
Afterwards.
That was a good, just absolute fiasco, ego death. But you know, it was something about it that was quite useful. Well.
But looking for this perspective, isn't it funny?
Yes, it's very funny.
Did you ever imagine like you're gonna be a PhD student at Harvard?
I didn't. I also, you know.
But did you ever aspire to?
I have no idea. I think that I really did want to do I wanted to be in the position that I'm in. But I also just know now, and we can get into this a little bit more, once it does. It's a privilege to be at a place like Harvard or like any other really esteemed university, but at the same time, there's nothing inherently. You don't just get there and then magic happens. It's not like it's the same as every other place. And in fact, many people just get there and then that's it. You got to do work when you're there and you got to like do stuff and it's really hard. So like I am at, you know, it is an achievement in and of itself to get to a place like that, but there's a lot of arbitrariness to it and a lot of things that are out of your control. And I got lucky a couple times, but the main thing is I just met the right people. I had the fortune to be in labs of the right people. Maybe I was smart enough to like sniff out the right people to like get me to do what I needed. But after I graduated and had this debacle with my senior thesis, I didn't even have a job lined up. So in the US, it's a little bit different than for a lot of the European systems. England is a little bit different in my understanding. But in the US, it's fairly doable to do enough undergraduate research because of how many undergraduate research opportunities there are at many, many, many schools. It's actually pretty impressive, the amount of money that the US pumps into research, which I really just couldn't say is the most important thing about our system. And then some people just go right to PhD programs, and there's no masters, and this is the thing that's really different. And so if you are not, like I clearly wasn't, ready for a graduate, degree that takes about 5 years if you're in life sciences, or 6 years.
Did they teach you at undergrad level to, do researches and how to, navigate that even? It's like...
The thing is, even in the US where it's like a very like well-established thing, like we have a lot of scientists and a lot of universities and a lot of institutions, but I didn't understand what a scientist or what a research path was until like I kind of just bumped into enough people that did it. And my parents certainly aren't scientists, and you don't just meet, maybe like your one friend's like uncle is a scientist, but like they don't make it clear what research is. Most people who think about science, or biology, who are 18, think medicine. And so it took me talking to a friend who was in a lab at my university, Brandeis, in order to then talk to more people. My entire first year was just figuring out what they do and actually what the path was. So they don't really teach it, but you have to find, and this is true everywhere in the world, you just got to find the right like mentors and the right people to tell you what the best path is, and to provide some optimism and tell you that you can do it. It's really like kind of the only thing that you need. You got to follow the advice and you got to like do the things, but that is how you know. And you talk to the professors, you talk to the people who are doing the research.
Let's talk more on your research, actual research that you're conducting. So it's on dreams and the science of why we sleep.
Yes.
And we don't have many answers,
Correct.
Not just in terms of why we sleep, in terms of our brain and how we are using it. We have some answers, you told me, in terms of senses and some other stuff, but for most of it, we're just, we're like, we don't know.
Yeah.
It's just a blank space. So.
Right. And this is especially why I definitely don't work on dreams, because there's no testable hypotheses about how we could understand the biology that underlies dreams. That's about as complicated as it can be. And so just to go off a little more from what you're saying, we don't even really have a good understanding, like a physical explanation for how the brain allows for us, I was going to say something even simpler. We don't understand how the brain allows us to breathe. There's like a small little brain region that we have a like decent understanding that is probably the center where breathing happens, but we don't know the mechanisms, which ultimately aren't at the neuron level. They're at a circuit level and a molecular level within the neurons themselves, how they organize this pulsing thing that you could then also, this is automatic, but then also I could choose to breathe, and how you get a both automatic and a controlled component to it. We don't even understand how that works. We have some ideas, but if we can't do that, how are we going to be able to understand, get a physical explanation for how we do anything, let alone intelligence or imagination, let alone those really or language, even very basic innate drives and motivations. And that is why, at least, I choose to study the things that are like, I like the innate behaviors that we have and the motivated behaviors that we have, like sleep, something that is just so impactful for our lives. And I think that if you study these things that are like, pretty much every animal does it. Every animal that has ever existed has a series of motivations. It's how you navigate through our day. And our behavior is really flexible. And so the goal for me is to study those types of things in the simplest system that we can. And so because of that, because humans, at least from my perspective, are really, really hard to study, for doing experiments on humans, There's two types of ways you can do experiments about the natural world. You can either break things and you can see what happens, or like, it's like breaking a gear on a watch or something and seeing what goes wrong. People use that example too much. Or you can observe things. You can like astronomy in the old days, and you could make correlations and make. You need both of these things, observational things and kind of manipulations, and you use both of these things to make a really good case for this is how something works. And so because of that, I don't want to use humans. We have tens of billions of neurons. We don't really understand how things work, and you can't do the types of experiments I think you need to do. So I choose to work on a model system. Mice are a classic example of a model system. They do a lot of things. They're mammals. And I work on flies.
Yes. Drosophila.
Yes. So I choose fruit flies. Fruit flies definitely do sleep. And if it's not sleep, it is something that is so close to sleep that let's just call it sleep. But I would absolutely argue that they sleep. And my lab, the lab of Dragašević, who I guess was on this last year,
yes.
But the lab studies sleep in general, and not just sleep. We study some other things like our 24-hour like circadian rhythms that we have, and some of the behaviors associated with that, of which sleep is an obvious one. So the lab kind of asks a lot why we sleep, which is a big question. In other words, what goes wrong when we don't sleep? She spent a lot of time talking about that.
Yes, so, with their gut.
With the guts, things go wrong to the guts, and it worked really well. They sleep deprived a bunch of flies.
Yes.
And by doing that, it's a really extreme system, and it's really extreme manipulation. They were able to find something, and then that made predictions about what we could then study in mammals, and it turned out to be true in mice as well. And so that's a good case study as to why using modeled organisms are useful. This has happened many times for fruit flies and making important discoveries. But I don't ask why we sleep. I'm asking a little bit more, I guess, how we sleep. It's not really exactly right, but more of the like mechanisms that underlie this one specific aspect of our sleep, which is that we have a drive to sleep. We want to sleep, we get tired, and we have...
But based on what?
Well, there's two ways to think about that. There are kind of things we could use from our own experience. This is why I like behavioral neuroscience, because unlike a lot of things in the natural world, you can kind of just use your own experience to like get some intuition for the types of inputs. So we know that time of day is a major input, circadian time of day. It's nighttime, sleepy, daytime, we're awake. And we know that there's other factors as well that are pretty obvious. Like if you don't sleep, if you pull an all-nighter, which I have sadly done many times, deeply ironic given what I study, but then you're just gonna sleep a lot more the next day. And it's not, you're probably gonna be tired throughout the day, fall asleep earlier. And then there's also just other things too that like, our day-to-day lives and our physiology demands different things from our sleep drive. Like, if you, depending on whether or not you're really full or haven't eaten like in like 2 or 3 days, whether or not you're sick, you tend to sleep more, and whether you physically exerted yourself, there's just a lot of little factors that pile up together to create this drive to sleep. And it's really important that our brains allow us to sleep and support our sleeping to the right amount, because we don't want to sleep absolutely a very small amount.
The right amount. Everybody keeps telling us that it's like 8 hours, 9 hours, 10 hours.
I think a lot of that is really hard to claim. I probably should know a little bit better as to the very specific reason. Everything is operationally defined in research, and that gets kind of forgotten a lot. That they say we are recommending 7 hours because of this, this, and this. Now, I just think in the reality of it, every single person is different, and every single age is different. I'm certainly, I'm just talking about other people's research at this point, but we sleep very differently when we're babies. We sleep very differently when we're 5 years old, our age, and then when you're also in like your 70s or 80s, your sleep drastically changes. Whether or not you need the same amount of sleep is a different question, but certainly the patterns in which you sleep and the amount that you want to be sleeping or how tired you are absolutely changes throughout life. And I think that every individual is different. The same way that every individual, if you're talking about like food or hunger, there's no like, one, like everyone has to eat 2,000 calories a day. That would be ridiculous. Every person has different needs. And it's a little more mysterious with sleep, but we all have different physiology, and there's different circumstances in our life, I think, which require different amounts of sleep. So the question that I'm trying to ask is, at least in the brain, what is it? What is that, like a physical representation that like really accurately explains what it is that is this like sleep pressure, the thing that builds up when we're really tired, that builds up through all these factors. And it might be several different signals. It might be one signal. It might be in multiple places. It's really super mysterious. And that is something that I think would be really a huge, like if we could gain some insight to that, I think just existentially that's really cool and important and like so relevant to our lives. And also, you know, we have sleep problems as a society.
Yes.
And, you know.
What do you think about those medicines we use for sleeping?
So again, I probably should know a little bit more before I say the thing I'm about to say, which is that we don't really have proper sleep medicines. Nothing is really we don't have a button that says go to sleep. You know, like without, you know, something like alcohol, like you sleep more, but that's not like really good sleep, you know, or accurately, you know, what you should have. Then there's like melatonin. Doesn't work in everyone, doesn't put you to sleep. You can wake up. If you take, you know, any sort of like sleep, like a cough syrup or like a NyQuil or something.
Oh, I did that in America.
Yeah.
And I slept for 4 hours and I got...
Only 4?
Yes, but I got dizziness and it was awful, and it was such a drive to just go lay down, and then I woke up 4 hours and
and that was it.
I didn't even know. We don't put that in cough syrups in Serbia.
It's, you know what it is? It's most of the time it's an antihistamine, like Benadryl.
But why do you put that in the cough syrup?
Because it makes you sleep.
Do you presume that also when I'm coughing, I also need some rest?
No, it's usually used as it'll say like a sleep aid. And it does make you tired, unquestionably.
Oh my God.
And this is a little bit how we know some of the things that contribute to our sleep drive and our sleepiness. And also how we know that it's not just gonna be one thing. We know that from antihistamines, very simply, that it makes you a little bit tired. A lot of people have that effect, especially the first generation of antihistamines, like the one in Benadryl, makes you tired. So we know that histamine, which is something that is naturally produced in the brain, plays a role. Then we know on the kind of other end that caffeine keeps you awake. And that is what people think, presumably through adenosine signaling. And that is something that then makes you not sleep so much.
Is that true?
Coffee.
In terms of coffee, yeah.
Yeah, but it's a
not for some people. Some people take it at like, 11 p.m. and they're like, no, I can do everything now. It's like a superpower after the coffee.
Yeah, well, I could drink an entire cup of coffee and fall asleep like no problem. Which again, it's like, but it's very clear. You take some coffee, you'll be a little bit awake especially if it's your first cup a day and you're not drinking that much of it over your like week or something. It has a obvious stimulating effect. And so again, with some more experimentation, we know that through the adenosine signaling, pretty sure that's right, that it has some contribution. So again, this has some contribution, histamines have some contribution. Another really good example is this is a good success in our understanding of sleep and sleep circuits, but doesn't tell us everything, is narcolepsy. So narcolepsy is a disease of which we really have a very shockingly good understanding of. Narcolepsy, is very classically associated with kind of spontaneous sleeping. It's not really the most accurate definition, and I'm not going to try to give the most accurate definition, but it is certainly an immense tiredness that exists throughout the day. And there's also this state of, if they get a little bit too excited, they kind of have a bit of a paralysis. Yes. And amazingly, this is why model organisms are great, there was in 1 year, 2 sets of research groups using 2 different systems found a perfect model for narcolepsy in different animals, and it was because of the same exact signaling pathway that got messed up. And they found, this one French group found a dog. They were able to selectively breed a dog that had narcolepsy, and they found out that there was one that had narcolepsy. And like if you see videos of it, it's really cute but really sad. There's like a dog runs down the hallway and then he just collapses and passes out.
I've seen those.
Oh really? Yeah.
I've seen those.
It's compelling.
I sometimes see my dog has that.
Yeah, it's really compelling. But what it is they found that it was a gene that called orexin, is like the popular name for it. It has 2 names. There was a mutation in this gene called orexin, which is a neurotransmitter. It's a neuropeptide that's released from one small place in your brain. And in this other group found a mouse, they made a mutant mouse that had a mutation in a different part of that pathway, in the receptor for that pathway. I might have had those 2 groups mixed up, and those mouse were also narcoleptic. And then they found in humans, they said, oh, maybe narcolepsy really just is because of how oddly conveniently specific the phenotype is, and it's so conserved in these animals, maybe it's what's going on in humans. It's not exactly what's going on in humans. It's not that there's a mutation in these genes. It's that the neurons die and it's an autoimmune disease where just those neurons die that have orexin, and that causes it. So my point is, you have all these different things that contribute to sleep, all these different molecules that we get a sense of. You know, the dopamine's important because of things like amphetamines that affects dopamine. These all have effects, but none of them together, if you mess around with any of these signaling pathways, none of them just force you to sleep. None of them are the sleep button, which is why a lot of our sleep medicines aren't really sleep medicines. And which is why this would be important.
Do you think that the lab that you're working in with Professor Dragana can lead to maybe a medicine that can help us mild the conditions of not sleeping?
My first hope is that we come up with an accurate understanding of some of these signals that really accumulate throughout our day and throughout our different conditions. Then we could try to make a drug from it. But those are, in theory, two different things. But if we get a really good understanding of something, then a drug could come from it. But I'm not a pharmacologist. So the first thing I do is just try to understand the basics of the brain. And then, yeah, it of course suggests that, like, this is how things have worked in the past. You understand a system, and then you find a drug that works. Or, you know, we just gave a lot of drugs to a lot of people for many, many years in old times, and they work for reasons we don't understand. And those are two completely different pathways to making drugs that ultimately try to help people and affect us.
And what about the learning and cognition? That's your part also, the memory.
Yes. So I have two completely different projects. One is on sleep, and one is on learning and memory. And obviously, sleep is something in which we have a lot of, it's really important for memory and really important for retaining information and for recall later. But my other project is, I think, we don't have really, I'm going to sound like I'm a robot just repeating the same things.
We don't have the
really
answers.
True.
No, but people need to know
this is important, yeah.
Yes, people think that science is something like you come to research and then you take 1 or 2 years doing that and then poof, that's it. We have a miracle.
Yeah.
Maybe I think it didn't even happen in the past. It just presented to us like that. Oh, the apple fell on Newton's head and then he thought that, you know, that is something amazing. Is the stories we're being told about that.
And even that's not even really at all what it was or how it happened. It comes from like one dude's story, like who was talking to Newton when he was much older. It was
yes.
And it's not, you know, he didn't just like, I'm going to understand calculus and use that to help explain gravity. And so there's that aspect. And there's the other aspect, which is a lot of people in science communicators, I think, falsely, whether it's their fault or not is another question, falsely claiming that we know more than we do. And we know so much about physics. Like, it's crazy. We know so much about chemistry and so much about biology, just, you know, and all of these things advanced over the last 400 years. So much we
don't know still.
And so much you don't know about the brain. And of course, you know, everyone's like, it's the most mysterious thing. And it is. I'm not like glorifying the brain. And I'm not like, it's so hard that I want to understand it. I just like that it's like something that's tangible to us. And that's why I like studying it. But the learning and memory is, we don't have a good understanding. Sorry, I'm just gonna talk. Fine. You gotta keep me on track. Yes. This is all my friends get annoyed at me. So we don't have a very good, I would say, molecular understanding of, like, there's a lot of different types of memory. There's kind of the
short-term, long-term.
Short-term, long-term, episodic, memory, piano.
What's episodic memory?
Probably what most people think of as memory. I remember that I was in a place at a time.
I parked the car there.
I specifically, where I parked the car, or like something that you could kind of imagine, I think is a reasonable definition of that. But then there's things like playing the piano. It's not really episodic memory. You learn something, it ends up being a more motory output. It's a little less, I'm remembering this thing. It is more of a learning process, but it's memory. You know what's
intriguing? Like all the draws you put there. It's like this goes to long term, this goes to short term, this goes there, and this goes there.
Yeah.
Like, how is the brain sorting out all of the info, not just throughout throughout the life, but throughout the day, the seconds we're talking.
Yes.
It's already collecting in such a high speed.
Yes.
And processing and already putting it in our memory.
And we truly have no idea how that works. I mean, we know, like, kind of some things, like, again, really dumb, big experiments. And dumb isn't the right word, but really, like, simple things. Like, for example, there's a very famous patient HM, who, those are his initials, which is how they did clinical stuff. He had epilepsy, really bad epilepsy. When in about the age of 20 or something, they took out his hippocampus because that was something that ultimately got rid of his epilepsy, but then he could no longer form long-term memories. So he lived his life kind of like the movie Memento in about 2-minute chunks. So that's how we know. We know that seems to be the place, and he can remember everything from before the surgery. So it's one of the ways, and this is a classic way, again, You could either understand something by making observations about the way things naturally happen and making correlations and making manipulations. This is a perfect example of a manipulation that sadly had to happen happened for his health reasons, and it was a surgery. But from that, he could remember everything from the past. He couldn't form new memories. What does the hippocampus do? From that experiment, it tells you that it makes memories, some types of memories. He can still learn other stuff. He can learn like some games, because memory is a distributed thing. You learn different things through different mechanisms, different places. And there's not just one way in which cells, the question really boils down to the biological system. How does a experience change your neurons, your circuits, your brain to ultimately lead to a future difference in response? And it's one of the most critical things that the brain does. So William James, the kind of like father of American psychology, he wrote a book called The Introduction to Psychology, and he was the first person to claim this word, to define this word plasticity. And I think people think about the brain.
Neuroplasticity.
Correct. Now, neuroplasticity has taken on its kind of own very specific meaning based on experimentation and based on some particular models. But I kind of like going back to the original thing that this guy said, because one, it's good not to stick to too many frameworks.
Essentially, what does plasticity mean?
What he says, it's a little weird, way of phrasing it. But he says that brains, or like any system that can have an experience and have a change, has to be plastic. And plastic kind of refers to this like band of usefulness of, think about it as like a material is how he describes it, where it's like, it has to be, I wish I could remember the exact quote, but to paraphrase, a system that is useful and that can actually learn from experience is plastic when, it is soft enough, when it can be molded by the world, and it can change and slightly deform the like physical nature of this substance he's talking about.
Can our experiences change the brain in terms of?
Oh, of course, for sure. It has to. Again, everything in the brain, it's not magic. It has to come down to some physical change, whether that be from literally the molecules within neurons that change the responsiveness to some things, or quite literally in the structure of the neurons, which a lot of people focus on. And maybe it's, it has proved useful, but it is probably not the only way in which neurons change their response properties or areas of their brain react to things. And so you want to be able to change, let's say like physically, and that's one thing you need. It needs to be soft enough to respond to the world and make a change. But it has to be hard enough that it doesn't change altogether. They have to retain this kind of perfect amount, because if it was too soft, you would overlearn from everything. And not to, this is a very specific analogy that I'm going to draw, but imagine if you just learned incorrectly, like you overlearned. I mean, so a post-traumatic stress disorder, and this isn't a learning and memory thing necessarily, but it is a forming associations that have become inappropriate. So post-traumatic stress disorders happens when there's some really bad, usually very acute event, often
war,
trauma, and instead of then forming kind of normal fear and anxiety responses, you then start making associations. This is one aspect of a complicated disease and a condition. You form associations with things that just happen to be around you at the time that it happened. And you don't want to form associations with like a bicycle that was near you when like a bomb went off. You don't want that, because then every time you maybe see a bicycle, you get really scared and have a bad like fear reaction and anxiety reaction. But that is something that can happen if our brains were, for example, like overlearning and overly plastic. And you also need to learn from experience. If you just, don't learn from like putting your hand on the stove every single time, you're going to lose your hand over time. Yes. So that you need to have your brain in kind of this right amount. And I study, again, in fruit flies, a very, very simplified version of a time in which a fly can learn something about its environment. And I think that we found some neurons that, I don't think that these are like neurons that we have, but I think that this is something that is an inherent capability of the brain, that there are some neurons that, depending on if I activate them around the time of an association, which I can talk about if you're really interested, or inhibit them, I can make them overlearn. I can make them learn way too much. I can make their brains too mushy.
You're manipulating that.
Yes, or if I do it the other way and I activate these neurons, then they don't learn from their experience. And so, yeah, that is essentially, I believe, might be something that is really inherent to brains, where the kind of interesting thing, and getting super technical now, is that, animals we have these big brains with these big networks, and we have some neurons that are very dedicated, especially things that are kind of responsible for like the doing of the behaviors. We have the kind of thinky parts of our brain, which people think of as cortex, but then kind of like the more older, like
reptilian brain.
Yeah, and like I don't like using words like that, but like it's just helpful, you know. It's not really accurate, but like it's just, you know, it's useful for this. Is that things are a little bit closer to complicated motor output. So things like in our hypothalamus that regulate our hunger, and regulate our sleep. There's a lot of places around there. It's like, you know, here-ish, and there are circuits, networks of neurons in those areas that kind of seem to make a lot of decisions about what type of behaviors you're going to do and seem to kind of control the behaviors. And there's parts of the brains where things are way more distributed, and it's not like one part of the brain does this. Like this is memory, this is, you know, intelligence or something like that. But within those, and so we have, you know, in the fruit fly example that I'm talking about, there is also a circuit that controls a very particular behavior. And the neurons that I was talking about are actually directly normally important for the behavior itself. And this is, you know, you need a bunch of neurons that do a bunch of different things to help create a behavior, you know, whether it's a decision-making thing or initiating a motor action or something like that. But it's a completely different role that we have found for these neurons. And again, I can talk a little bit more specifically and less abstractly. It's a very specific role that these neurons normally play. But now we found that we can mess around with it and tune it, and that can vastly change how the animal learns from their experience. And it might be responsible, like I said, for kind of keeping this part of the circuit that kind of perfect level of plastic. You could make it so that they're like overly mushy.
Do you think it will be possible with humans to manipulate that?
No, well, there's two problems here. One is this in theory, if I can...
Is it applicable to humans at all?
Well, I think it will be. I just, you know, there's two. Is it applicable at all in the sense that if I could magically do the thing in the fly, but in the humans, if it was in the right place, if that type of circuit logic necessarily exists, I think it is a prediction that we can now make, and you could test it in a more complicated system. We do the hard work in the fly, and someone can do it in the mouse, and that's essentially a mammal. It's close to humans. Then there's the other part of this, which is will we actually be able to make a manipulation that is so specific like that? In the human, that's a whole other conversation. This gets into, I think, kind of the nature of more of my day-to-day in my research, which is what we can do in the fly. You know, in humans, our technology is pretty much like EEG, have a human do a thing, do you see changes? fMRI, have a human do a thing, or have a disease happen, see if there's changes. In the fly, we can do so many manipulations. In fact, you know, the fly's brain nervous system has about 100 to 150,000 neurons in it, and it's somewhere in that range. And we kind of know all the connections, and knowing all the connections absolutely does not tell you the brain. Like I said, we learn from experience. Things change in the brain. Obviously, like a snapshot of all the connections isn't 100% right and doesn't tell you necessarily about function. You could do this other thing, which is we have the ability through magical genetic tools that I didn't create, but I just get to use, that you can manipulate pretty much any single neuron you want in the fly brain in any way that you want. You can, you know, play
with neurons of the flies. What an interesting job.
Yes, it's quite nice, and it feels like
poking and waking up the fly.
It feels quite boring when you're actually doing it, or trivial. And like,
it looks exciting to me.
Oh, it was quite exciting, but like any person can come in, and so we take a lot of undergraduates in our lab, and I think that, like, you know,
they can come and see.
They can come and see, like, just like if you want to work in the lab, which, you know, we can't just take everyone and cost money and stuff, but like the first day in the lab, they can do these really complicated experiments. You don't have to set anything up. And so one of the ways in which you can manipulate, so there's a lot of ways in which you can manipulate cells in the fly to study them. You can, and I'm going to kind of gloss over some things here. So you can turn them on and off, and you can do it conditionally through different tools, the most famous and recent of which is called optogenetics. Opto meaning light, genetics, actually not a useful terminology in my mind to describe what this is. But what you could do is you could take neurons, so fly's got 100,000 neurons or 50,000 neurons. Let's say we can target 20 of them. I'm not going to get into exactly how we could do that, but it's very, very simple. And you can make these neurons express. They don't have this thing, it's a gene, you can put it in there, and now they express this channel. It's an optogenetic channel. And this channel, neurons are, of course, really care about your membrane potential and care about, they have electrical potential, so there's a difference across their membrane, and it is when enough depolarizations, and the membrane gets excited and excited through ions, through molecules with electrical charge. That when it builds up, you eventually get across a membrane potential threshold, and then you get a action potential or a spike. People think about spiking isn't everything in the brain. Not all neurons spike. Not all neurotransmitter release is spiking.
Means what?
Exactly this very specific thing that I explained, which is on a graph. You can see that, like, you put a little membrane potential in, you make the neurons a little more positive. They start off negative.
Do we have spikes?
Do we? Yes. Oh yeah. It's definitely like a fundamental aspect of many neurons, and it's important. But it's like you can make the neurons get a little bit more excited, but it'll just decay away, and you won't end up getting neurotransmitter release, which is the key at the end of this. It needs to communicate and release something at the end. And what a spike is that once it reaches a certain threshold, some specific voltage-gated ion channels. They care about the voltage. They recognize, oh crap, it's at this exact voltage. I now open up, and you get this kind of runaway process that's automatic and will always be exactly the same for an individual spike. It goes up and it comes back down. And that then goes down the axon of a neuron. Your typical neuron, of which you've seen, they're not all like this, but doesn't matter. You have like a dendrite where you receive stuff.
This one with like beautiful looks like a flower.
Yeah, it's they don't all look like that, and it's pretty important to know that. That's like it's usually
it's biased.
Oh, for sure, right, and it's pretty, and it's usually either hippocampus neuron or a cortical neuron, and they have those really crazy patterns, but like they don't all look like that at all. The ones that
like ugly.
Some of them can be. It's all pretty. I mean, there's
snakes.
They look like they have weird morphology. Like all cells are effectively just cells at the end of the day. Neurons have some kind of funky biology, but they do the same things as other cells as well. Anyway, I totally and completely got away from the original point of talking about action potentials and electrical excitability, because what I'm trying to say is in our fly, if we want to activate like 20 neurons, one neuron, we express this ion channel, which can pump in positive or negative ions, but it'll only do it when there's light of a particular wavelength. And so you can make it so that you can activate a neuron just by shining a red light at the fly.
Who found out that?
There was a series of people who both...
And how long did it take them?
This is a whole story, and will probably be it is a bit of a contentious debate. So the idea of it was, people thought about it. There was a couple ways to do it. And one guy developed a way, and it was really important. It was like the first tool, but it was not as efficient as what I'm talking about. But it was essentially a conditional way to turn on neurons through a light, but it wasn't through this channel. These other people, the way it was found is that, for anything that you want, you just look to nature. If you want to do something impressive to biology, chances are something kind of crazy in nature already happened. Lots of our tools have come from weird little findings. So the original light-sensitive channel that was found was in I believe it was an algae, or it was a single molecule. It was a single-celled organism, and that they care about light. Algae needs to photosynthesize, and again, I'm pretty sure this is algae, but I might be wrong. They need to photosynthesize, and so they care about light. And so when there's light, this channel opens up and makes them go in a place, and when there's no light, they don't go in that place. And so they found that channel. They then cloned it, the gene for it, and then put it in another animal. That was a different group that did that, and was able to then elicit spikes. Yeah, it's a bit contentious actually, because the guy who found it originally doesn't get as much credit as he should. It's a whole complicated thing, and I do not want to get into that. But I don't know any of these people personally, obviously. So, that ended up being a really useful tool. This was it's become in the last 10 years very practical to then actually implement this in our research. And again, I get the benefit from that.
And where is it leading you?
Well,
these manipulations.
The ability to make these manipulations can then teach us something about the brain. So that's one thing you could do. Another thing you could do is you can just delete or perturb any of the 14,000 genes. So
no, genes. I was talking about what if you can perturb the memory?
Well, ultimately that is what happens with us. So we have these I could explain the behavior now, and I'll try to be simple about it. I don't know how much time we have or how much time
we have time.
Great.
We talk.
Great. And you can edit. Whatever is boring. So, again, I study fruit flies. It's not because I love fruit flies or I just think that they're the smartest things in the world, but because there's enough of an element of truth in their behavior, in their nervous systems, in their biology, that we can learn something. from them. So, fruit flies, this is going to sound strange for about a minute here. Male fruit flies perform a very special courtship dance when they want to mate. And fruit flies have many drives, like ours. They want to sleep, they want to eat, and they want to mate. And this is something fundamental across the animal kingdom for things that mate. And they want to do it. And so much like pretty much any like David Attenborough special that you've ever seen, like peacocks or like birds in the wild, or a lot of things have a courtship ritual. Frogs, they make their noises, crickets. These are technically all forms of like courtship rituals, which are trying to elicit an excitement from a female who will allow you to mate. And so the male, it's really cool, it's very clear when you see it, is that they perform a song. They dance around the female, they follow her around, and we have these little chambers that are really easy to make, and you can put the flies in there.
They
and
do that.
And so
it's like a bowl for flies.
It's exactly what they do, and they sing. And so it's a wing, and they stick one wing out, and they chase the female around, and he pulses it in a certain frequency that female of that species likes. There's a lot of different fruit fly species. There's a lot of different types of flies, all very similarly related.
So interesting.
They have, and the females specifically like that frequency, and the males sing at that frequency. Some species of these flies have slightly different frequencies, and the females from that species prefer that frequency. That's how you get speciation. That's how you get other species that don't mate with each other. Fine. Now eventually they mate and whatever. Importantly, these flies don't necessarily, I think it would be advantageous, and I am making this up right now. This is important to make up because there are things about the world which we can hypothesize about, and there's things that just you say. And this is one of those, which is that it's probably not very evolutionarily advantageous for the male to waste his time on something that isn't going to bear him progeny. So not every fly in the wild wants to mate with him. If it's a fly of another species, he might try to court her, and she will not want to mate with him. That's a waste of his time when he could be mating with the proper fly for him producing progeny. So there's other circumstances, not just across species, which is that males also, female flies, a virgin fly is always very receptive. But a mated female fly.
How do you know it's a virgin fly? Or do you like...
We make it, so that's the case. Yeah, you do it by age. They don't mate when they're really young. And so if you could tell if a fly was like just...
A virgin fly,
yes. I told you it was going to be really weird for like a little bit, but once we get past it, there's neuroscience there that we do stuff with. But the way it works is that we take flies when they're very, very young. We know that they're virgins, et cetera, et cetera. Actually, we can also just kill off all the males by putting a gene on their Y chromosomes that if you make it warm, they just all die. That's how we make these things in mass, so I don't just waste my time. But we call it a virginator, which is, boy, just an awful phrase. But it's how we say it in lab. So anyway, a male will court a virgin female. The virgin female will be receptive. But if a female mates, she actually, kind of strangely, not really what you might imagine, uses the like sperm from that same male to fertilize eggs for like over the course of a week. And so that means she doesn't really need to mate again to lay eggs, fertilized eggs over that time period. And so it's not 100%, but there is a large tendency for virgin female flies, when they mate and become mated females, they'll be completely unreceptive to the male. And
they didn't look at him again.
Well, and more impressively than that, the male, we're almost at the learning part. We're almost at the experience dependent part here. If the male doesn't know any better, he will try to mate with her, and she will actively reject him. She will kick him in the face. She will like run away. The other female will like slow down, like maybe think about it for a little bit, and then this female will kick him in the face and run away, no matter how hard he tries. And so males that don't have experience in the way that I'm about to explain it, if they grew up completely without any social interactions,
with the females,
with anything, and I'll explain in a second, they will try to court and try to mate with a large variety of things. Not everything. They're not going to try to mate with like this bobblehead thing of Einstein. They're not going to try to mate with like a ladybug necessarily, but they will try to mate with some flies of other species. They will try to mate with virgin females. They will try to mate with mated females, and they will also try to mate sometimes with males. And only one of those things, a virgin female, is going to provide them the opportunity to have progeny. And if he grew up without this experience that I'm about to explain, He doesn't want to court. He'll kind of just court everything, mostly indiscriminately. Virgin females are still the most exciting to him. But if I do something bad to the fly, I make it really, really hot. I like bang the chambers up and down. I give them an electrical shock. While they're courting, or over the course of like a 30-minute essay, or at the end of it, I do this bad thing. Afterwards, he doesn't court all of these other things. He will only court virgin females. So then he learns to, for some reason, to avoid all of these other less good targets. So what we are studying, and he'll still just court the virgins, what we are studying is how is it that and this isn't permanent. This will go away over time. You don't want to have too strong of a learning where you just like, maybe it's still useful to give it a try and try to court in the future. But what he does is that this will go away after like several hours. You can make it even longer if you want, make it upwards of a day. You can make the memory stronger. Eventually he'll go back to trying again, but then he'll learn again and the same process will repeat.
And he remembers?
He remembers for a time period. Yeah, it can be in the kind of typical assay we do, about 5 hours, but we can get it upwards of still in effect like 24 hours later if we do the right set of things. And so, again, it's, is this learning how to play the piano? No, of course not. It's not, nor is it like a really complicated, I remember like a very specific memory of like how I drove to work in 2017. It's not that. But what it is a very obvious experience-dependent change in the animal in which he has an experience, something happens, he has a very specific response as a result, and importantly, it's temporary. Memories can be made and go away, learning can come, and the results of that can go away. So why on earth am I even studying that? One, it's simple. I think I've justified that it is learning a memory. But importantly, in the fruit fly, of which has 100 to 150,000 neurons, we have a very good understanding of the neurons, both from the sensory to the motor, that are responsible for the male performing this courtship dance and deciding whether to court or not. And so we have a good understanding. This is from work from a lot of it, some of the more recent work from Michael Quickmore's lab. This is husband of Dragašević, yes. Also a professor at Harvard Medical School in a different department, but there nonetheless, is that we, and many other researchers over the course of like 10 years, 15 years, it's a really good little group of research. The reason we're able to know so much about this circuitry that makes the male decide whether he wants to court or not is because out of these 100 to 150,000 neurons, the male and the female's nervous systems are, of course, majority the same. Both animals need to do the same types of stuff. They both need to see, hear, taste. They both need to eat and fly. But there's a small subset of behaviors that they both do that are inherently different. The male does this crazy courtship ritual. The female does not do that. The female will choose where to lay her eggs and has to have the ability to lay eggs. The males do not do that. So if there's some slight differences in the behaviors that are sex dependent, then that means that very likely, and it ends up being the case, that parts of their brain, small parts of each of their brain, are different or sexually dimorphic, which is the proper phrase. And this is true. About 2% of all of these neurons are sexually dimorphic. And we know that because it is just one or two genes, specifically transcription factors. Transcription factors are genes that will bind to DNA and drive transcription of often specific genes. This is an important thing for the regulation of why neurons are different from each other or why neurons are different from cells in my skin. And so these two little genes control these, mark these neurons that are different in males and females. And so now we've gone from a human brain of tens of billions of neurons to a fly brain of 100,000, 150,000 neurons to now 100 to 150 neurons. Sorry, that math can't possibly be right. And it ends up being about 1,500 neurons.
Yes.
I'm not a mathematician.
It's fine.
Yes. But it ends up being about somewhere, and this number kind of changes, and I'm not being super specific on purpose, about 1,500 neurons. They go sensory to motor. And so over time, we've used the knowledge that these are just small, and we can see where they are by staining for this gene, these sexually dimorphic genes that I was telling you about. And so, great. So we can access the small populations of neurons, and we know the exact place in the brain, at least that seems to be the case, of the fly brain that is responsible for generating this courtship behavior, for choosing whether they want to start courting, taking information from all of their senses, whether or not there's a female there visually, all the pheromones, their own courtship song, quite a few things about the female, and their own behavior that go to these one set of neurons that are called P1. They have a name. And the name's not important. But it's about 20 neurons in each of the hemispheres of the fly brain that controls pretty much every aspect about this like really complicated behavior.
Did you test that in mice?
Mice have neurons and groups of neurons similar to this. There's not too many things in which you can stimulate. So if you stimulate these P1 neurons, and I'm saying this now so I can explain what it would look like in mice and humans, but because this behavior is so innate and stereotyped that if you activate these neurons, they will court things that they don't want to court. They'll court a piece of rubber band if you activate these neurons, and they'll do all the parts of courtship. They'll try to mate with it. They'll do everything, and also, importantly, males will not want to court if they've mated a bunch of times. They lose their mating drive. They have a drive to mate the same way that many animals do, and it's because of dopamine that there's a small set of dopamine neurons that talk to this P1 neuron. They form a connection, and when these animals mate a lot, their dopamine goes from being high to low, and that makes P1 not care so much about their sensory, if there's a female there or not, so they choose not to mate. And so if a neuron really controls all aspects of like a decision making, or a lot of aspects of decision making and behavioral initiation, then maybe there's a case like that for humans and for mice. And it does seem like in some circumstances there are definitely dedicated circuits that really tend to do to generate behavior, kind of whole aspects of behavior. So there's like a really crazy and like pretty like well-known experiment now. It was one of the first like very popular uses of optogenetics, which you talked about earlier, yes, where they expressed in the lab of David Anderson in Caltech, I believe. Don't quote me on that if I'm wrong. Okay. And this woman who's not a professor, Dai-Yu Lin, they put these optogenetic tools through genetic expression in one small part of the brain of the mouse that people thought was important for aggression, for example. Yes. And to me, the behaviors, doesn't matter specifically what they are, as long as these principles hold up across these behaviors. And what they did is they took a mouse, they put this optogenetic tool in that part of their brain, they express this channel, and when they turn the light on, for mice it's different. You have to do a surgery and put an optical like fiber.
Device. Yes.
They turn the light on in this area that's supposed to elicit aggression, and the mouse, is alone with itself and a rubber glove. And it's a blown up rubber glove. And this video, you could find it. It's on YouTube. Yes. It's crazy. They go from not caring about the rubber glove to they turn these neurons on with this optogenetic tool, and the mice go crazy and just attack the glove. It's just a glove. And so my point is, there are neurons and groups of neurons that control the decision to make, to initiate a behavior and kind of just control the behavior itself. Not all of the brain is like that, but things that are very like innate behaviors, there are circuits that seem to be dedicated to that. And why I bring this up is that, going back to our learning and memory thing, yes, these P1 neurons that seem to kind of control this behavior, of which there are likely larger groups and slightly more complicated dynamics of neurons that control behaviors in mice and in humans, and this is true, we could see in like stroke patients, kind of like weird sense of behavior if the right part of the brain dies and gets a lesion. In flies, so these flies learn if they had a bad experience while they're recording, they don't want to court all these bad things afterwards. They won't try to mate with it. But if we do something very specific by turning on or turning off this one population of neurons that goes to P1, goes to this central behavioral circuit, the kind of decision-making point, we can make it so that if we activate these neurons just around the time of the punishment that happens, they'll court anything, you do something bad to them, they won't want to court the other stuff, they'll only court virgins. If we activate these neurons just in that time, then it can erase the memory. That's one thing. The other thing we do, which gives us a little bit more, I think, interesting stuff, is that it can go the other way. If we inhibit these neurons that go to P1, then that will then cause the males to have super learning, just like making the brains like too mushy to the fact where they learn things that they shouldn't. And in that case,
that would be amazing in humans.
I don't know if we want that necessarily. Maybe in some cases.
Like Limitless.
Right.
Movie Limitless.
Yes. So if we go from using 10% of our brain to unlock 100%. If we unlocked 100% of our brain, we would just have a seizure. It's totally bad. There's specific things you could do.
Is there research about that if you unlocked it?
There's no unlocking. It doesn't make any sense. Do you want, our neurons control stuff, you know what I mean? Like, it's tied to our bodies and our realities. If you just activated everything, it's gonna not make you happy. Our brains work because of not everything is active at once. That is so critically important. You could do it. You could do it. If you express these optogenetic channels and every single neuron on the fly and they just go. They do. They have seizures. Dead. They have seizures. Yeah, and they'll pull out of it. But like, yeah. Okay, so if they make them super learners, they will learn not to court these virgin females too. They will stop courting pretty much everything, and that's like pretty shocking. And it also will make them learn to like really gentle things. So instead of making it like really, really, really hot, like when I do this like this heat thing to the flies, where I give them shocks, they'll learn to like a little tiny amount of heat or a little tiny amount of shock. And they're kind of learning too much. As going back to the William James analogy of plasticity, it's making their brains too mushy. You don't want that. And so what the key, I think, finding is going to be, we're still looking for the mechanisms and want to get some more insight out of it.
Yeah. Where are you getting at?
Is that it's like some neurons that are normally responsible for controlling a behavior, something that is built into the system that has a normal function in the doing of a behavior, of which we do many all the time, and our innate behaviors is how we spend a lot of our days and the things that we revolve our lives around, that neurons that are already built into the system can regulate how much we learn from our experiences in a way that is specific to the particular behavioral circuitry that we do. You know, a lot of memory is completely decentralized, hippocampus things, and
in a lot of places.
It's in a lot of places, and it's not like, you know, again, an episodic memory isn't necessarily related to a particular, how you want to change a particular type of behavior, but for this type of experiential learning that affects a particular output, this might be something that this is where the memory can lie, and that this is something that if something goes wrong and this system goes out of whack and is not kept in check, then you either get like inability to learn or you learn too much, which both are inappropriate and not useful for survival. And so it's not just going to be like one neuron in one place. It just ends up getting more complicated. The circuits get more complicated as the animals get more complicated and you add more neurons to the brain. But there's still some core principles there that we could learn and have a much easier time studying in the fly's brain than we do in the mouse or the humans.
Right?
Oh, yeah, but like, oh my God, do we need those baby steps? Because otherwise, it's literally impossible to figure it out. And you could do this in mice too. And
in terms of years, how many years do you think you're gonna be analyzing flies and mice before you get to anything crucial that can be groundbreaking in terms of us?
Oh, I think the crucial groundbreaking comes from the flies. Whether or not you test these things in humans is a different question. It depends.
In your opinion?
Flies, I'll never give a proper answer. I just like talking. So fruit flies are responsible. I guess you heard me talk about this yesterday. Yes. You know, basic fundamental biological discoveries in fruit flies are the basis of 5 different Nobel Prizes that include how genes are arranged on chromosomes, fundamentals about how mutations, like X-ray can cause mutations in flies, about how our innate immune system works, about how embryogenesis happens.
And all those on flies?
All of these on flies.
And they were proven in humans later on.
There was enough principled understanding that it was then similar enough in humans where it was just correct. With slight modifications, slight idiosyncrasies, that's fine. In the 5th one in 2017, closest to home for me is, for actually multiple reasons, is the understood using flies and mice, but the groundbreaking work was in flies, understood the molecular and genetic mechanisms that allow us to keep our circadian 24-hour rhythms in which so much of our lives revolve around. And so those genes, you know, pretty much everything that we found there, again, slight idiosyncrasies in like the way that we found that how like some aspects of embryogenesis work, then like how you have molecules, you start with one cell and then you have some molecules do a thing, and when the cells divide, the cells have to turn into special things and go to the right places, and we have limbs and a whole directionality to us. That was found just by randomly mutating genes in flies, and just
long live the flies.
And really carefully looking at these things. And look, you would get like a total mess up of like a wing or like something organs in limbs that we don't have. But it doesn't matter because the genetic principles and the molecular principles are the same. And then actually the most impressive one is and how similar it was for how oddly specific it is in circadian rhythms. There's kind of like a core 4 genes.
We need to explain circadian rhythms.
Absolutely.
Yes. Yeah,
absolutely.
What are they?
Yeah, it's just, all of our times a day, you could probably associate personally and very tangibly with like a certain feeling or a certain set of actions. Sleep being the most obvious one, tend to sleep at night versus the day. And circadian rhythms are so consistent and always match this rotation of the Earth at around 24 hours, really closer to 23 and a half, that like if you fly on a plane to a different time zone, you feel incredibly out of whack, and that is usually one of the jet lag, and that is one of the causes of that is you're effectively changing when light is coming on and going off around your time of circadian, your own internal circadian clock. And we could do it importantly in the absence of light. That is the magic of the circadian rhythm. It is a set of molecules that is mostly by changing expression of some genes that get made, take some time to degrade, and it actually inhibits its own transcription and making. In this process, it goes away, comes back, goes away, comes back. It takes about 24 hours for that to happen. I really quickly explained the molecular mechanisms, a little more complicated than that, but the point is, the kind of core genes that control that, a lot of them are the exact same in humans, or 2 of them are the exact same in flies and in humans, but it works the exact same way. We have no idea exactly how those rhythms how those molecular genetic rhythms turn into our behavioral and physiological rhythms. I mean, our physiology also really plays into the circadian rhythms as well. We get hungry at certain times of day, we're more like aroused at certain times of day. These are circadian rhythms. But again, how we found these genes? It was random mutagenesis in flies. But the very specific assay that was used by this guy, Ron Konopka, who didn't share the Nobel Prize, but he was the first guy that found the gene called period, named so because what he was looking at, he wanted to see if we could mess up timing, to mess up these rhythms. And there's one thing that flies do, so much like butterflies, they start off as like a larva, and then if butterflies, they form a cocoon, and they come out, they eclose. The term is called eclosion. Flies do a similar thing. It's not a cocoon, but it's called a pupa. And once they stop being a pupa, they emerge from this case. That is, they usually do it in the mornings, pretty consistently. And
in the morning, they become those.
They pop out and they go, I'm now an
always
adult
in
fly.
The morning.
It's pretty, not always, there's always like some, but like if you look at the distribution, it's like peaks right at the morning, every time. And of course it doesn't matter if it's literally human.
It's like waking up.
Yes, I will be very careful about, I wouldn't necessarily call that sleep because they go through metamorphosis, which, oh my God, I don't really think that's sleep. But importantly, the guy just made random mutations in flies and found that he found some flies, once he made a mutant, one type of mutant, they didn't wake up in the mornings, but their rhythms were like 28 hours. They had like long rhythms. And then there was also flies that woke up too soon. They had really short rhythms. And there was also flies that had no rhythms at all. They just woke up, they closed random times a day. No rhythm at all, just random times a day. And so those are 3 different genes that he found, and using some old-timey like gene mapping techniques, found they were all manipulations of the same gene. And this thing didn't just control when the flies closed out of their pupa case, but like all their other aspects of their circadian life as well, when they're most active, when they're asleep. And again, that is an oddly specific thing about fruit flies, where any person would be like, what on earth does that have to do with anything in the human? But if you take it at the right level of abstraction, you don't want to take it too seriously. You don't want to take it like, too not seriously. The right level of abstraction, you can learn, this is a mechanism for timing and sort of 24-hour rhythms. Maybe it's something that's important in humans, and it turns out is the same mechanism and very, very, in 2 of the same genes. The other genes effectively do the same thing. So the principles that you learn in these weird, wonky, and simplified systems are critical for our understanding of things that we can't do experiments in. Science is just about doing experiments in things that you can do experiments in. All hypotheses have to be testable and falsifiable. They have to be able to falsify and confirm. And if they don't do that, then it's not a theory. It's just you saying something.
What do you think about scientific research in general now? Considering all the factors you just said and the baby steps and everything you're doing, in terms of what we are hearing, sometimes it can be like an over-assumption and you jump ahead of a conclusion and you're still not there, just to please somebody, which can be good.
Or careerism. Yeah.
Yes.
Keep your job. Yeah.
Yes.
You know, in the history.
Is it hard to be a scientist? Nowadays?
I don't think it's hard. You know, I only know nowadays, and two, I almost go out of my way to be more optimistic than I think the average science person at my stage is, just because like people are so.
Is it this because you're young?
No. You find people like Dragana, like Mike, who are just positive about being a scientist is like such a pleasure and joy, and it's really possible to make discoveries, and it's not all about like, oh, it's so hard to get money, I hate writing, and it's so hard to like do this, and it's all like, you know, it's all like a political thing. It's you know, some of these things are of course true. Not everyone has the easiest, and it's a difficult job the same way all jobs are difficult, and there's special things that are unique, and it's hard to get money, and of course, but like, if you want to do it, you can do it. Like truly anyone can do it. I think a lot of people who are scientists, I don't view them all as like geniuses having a eureka moment.
I was just about to ask you that, because we perceive you as some geniuses who are doing that in their labs.
Oh, please don't consider me. I'm
God
quite
knows
stupid.
What, you know, and we have those biases towards that.
Yeah, of course. And you know, I do think that there's a little bit of the way in which some scientists act, especially a lot of science communicators, not the good ones, that they make themselves seem really smart by like saying words that are like really complicated, you know, and saying, you know, talking about things that just they sound like they know so like, you know, sure about, but you know, I do think that legitimately anyone can do it. Not 100% of the population, you know, but like I would say a vast majority of the population, if they wanted to and had, you know, it gave them a level of joy that you kind of need, you can kind of have anyone do it. And importantly, you need to have the right people guiding you.
Right mentor.
The right mentors. Science throughout history has been like, you know, science philosophy, pretty much any like time in which there was areas of study and rigor, and especially times when you use the scientific method, is just a history of everybody having like one famous scientist had progeny of another famous scientist, and
just
these
add
people,
up.
It adds up. And my point is like mentoring is just like so crucial in the world of science, because it's like, I don't know, you got to pay it forward, and like it's just the way in which you teach somebody, and it's more than just teaching someone. You're not just like teaching them how to do how to like take on a research project, and just teaching them how to like do a good talk or how to do a technique. It's really about giving them opportunity to have their own ideas and like to be independent, and it's a hard thing to be a really good mentor, but it is the most important thing. And I was very bad at doing science my first 2 years in research, and it was nobody's fault. I was just pretty absentee. But when I then realized, I obviously knew I couldn't apply to PhD programs after having my defense canceled and not even having a job lined up for a little while after college. That was a super unfun time in my life. But I eventually, after literally cold emailing over 100 scientists in Boston area.
That's so important.
Yeah.
Email, guys, email.
Oh, it's and I hated doing, and my emails were so bad. That's why it took me I just watched them. I read them recently.
There was ChatGPT back then.
Oh my God. I reread them and I was like, what are you doing, dude? You're so dumb and don't know it, naive.
Nobody's gonna respond on that one, right?
Right.
But somebody did.
Well, they did. And that was Mike, you know, and he, one, could not be more appreciative that he gave me that opportunity. But, you know, there's something that
you just need that one who wants to believe in you.
Yeah. And what's important is like, you know, is that they like give you an opportunity. You got to find the right, you know, because someone could hire you, but the job could be not what you need. I was looking for independent research experience because I knew if I was going to go and be a scientist, I had to figure out three things needed to be accomplished with this job that I had between undergraduate and PhD, which was, and again, there's no masters for us, so this is really where a lot of that post-college experience comes from for pretty much most people in grad school these days.
And it's for 5 years?
PhD?
Yes.
It depends.
Yeah. In your case?
It depends how the next year goes. 5 or 6, I'm in probably less of a rush than I should be. But it's,
you like it there.
Yeah, it's not. I don't like that. I should be faster. This is my bad. I should be getting through this faster. But I would like to come up with a couple good stories, both on this like sleep need thing I was talking about and this memory stuff, which is a little bit closer to being published. I want those 2 things to be done before I'm gone. Take some time. But undergrad, lab tech, or research associate, or whatever you call it, and then PhD. In that 3 years, I knew that if I was going to go on to grad school, or if I was going to be a scientist, I had to, 1, know if I liked it. That's not something you can do until you are given independence as a researcher, as a young researcher. And a lot of labs don't necessarily give you that if you apply for an RA job. You might be doing something a little bit more managerial, even though you can still have the opportunity to do independent research. Sometimes you're just stuck with somebody who's a grad student or a postdoc, and you're just stuck with them. And then there's the reason I took the job with Dragašević's husband, Mike, is because he said, I'm going to treat you like you're your own graduate student, and like you get to do independent work. And through that, you'll learn. I was very, very bad at it at first. I really didn't know what I was doing. but
that's the process. It's the beauty of it.
And that is why the other thing was I wanted the experience. And I wanted to, again, see if I liked it. I wanted the experience. And then that third kind of shallow thing is I needed some more years behind after my bad GPA that in my multiple failed classes.
It's such a motivational story.
Thank you. It's actually not. I was just if I just wasn't an idiot, that's not true. It's it was a lot of it was self-induced, but reflective of some deeper thing in my enjoyment or lack of enjoyment or difficulty in taking classes the way that they should be.
Yes.
Yeah, and I think that most classrooms and most professors are not good environments for most people, or they can be. Some students, they learn the right way, the way.
What's the way of learning things, for example, in science in America? Is it experimental, theory-based?
It's
or mostly ex cathedra, just professors standing and teaching?
Yeah.
Versus the one in the Ivy League? I mean, your university versus the Ivy League one?
Yeah. So you mean like my undergraduate?
Yes.
Not, yeah. Yeah. I think at the end of the day, and this is just me kind of operationalizing what you're saying, if you want to learn the material of what is known about science already, a classroom is fine. A textbook is better, honestly. Most teachers don't put the time of day in that they need to actually make students comfortable and to treat them right and have respect. I really do find that a lot of teachers don't have respect for students in their classroom in the way that they should. That wasn't my problem. I just was a bad student. But also, they just don't give them the time of day that they really need to teach a subject in the most interesting, important way and to justify it and then to then grade appropriately. It's an ununiform problem. And so if you want to learn material, that's fine. And a lot of people have learned a lot from that. And I am probably underrepresenting the amount that I have learned from the materials of some of my courses. But to be a scientist, it's not like the material is just so advanced or something. You have to be so smart. It's just different. It is knowing what we know, and then the act, and it's its own way of doing things. It's using the scientific method to uncover some discovery about the natural world. That is really what needs to be taught, but you can't teach that in a classroom. You learn that in a lab from a mentor, including graduate student mentors, including postdocs, and the ultimate one being the faculty member, the professor, the person in charge of the lab, who has done it many times, has done it for many people. They themselves were in the trenches doing it at one point. So that is the way to learn, and a benefit that a lot of American universities have that isn't so common everywhere else in the world is that, 1, we have a lot of universities in America, and 2, a lot of them,
variety of choices.
A lot of them do really good research. A lab is a lab. Harvard is special because it has a lot of money, obviously, and it has a lot of people that are drawn to it. So you get a lot of people and a lot of resources. It's easy to do stuff. But a lab is a lab anywhere. Discoveries can be made literally anywhere. That's not me trying to be like, everyone should just do it, and there's totally no disparity between like a place that like Harvard has all the money in the world and another place that doesn't. But the point is that in America, there's quite a lot of opportunity for students. Even they don't have to go to university. Say, oh, it's the summertime, I'm going to go do research somewhere else, and you get the opportunity that way. Or there's internship programs. And the point is, you just need to get in and do it. and do it for long enough to see whether you like it. Because then if you're doing a PhD for a bunch of years, it is not a fun time if you don't really enjoy it. It's very serious, like, amount of effort that has to go in to make a discovery. It's super fun and I enjoy it, but you know, it's not, you can't just, you know,
instant.
There's no, I've incepted some idea. It's a lot of just doing stuff, you know, whether or not it's, you know, at the level of like wet or bench work in which you're doing physical experiments or something completely computational and theoretical. Both things require a lot of effort to just produce something. Take some years, but like, we could all probably be a little bit faster, myself included. But you know, you can only learn from doing it and knowing if you like it or not. And,
do you see yourself as a professor, as a future professor, staying there?
So professor, I will break that word up. You can, so being, I would like to be head of a lab, have my own lab with my own scientific direction, making my own discoveries. When I say making my own discoveries, I really mean having a research program, but all of the graduate students and postdocs technically are the ones doing the research. But the professor is the one in charge of the lab. It's their work, you know. But there's that, and then there's teaching. The teaching thing, you know, I clearly am someone.
Are you good at it? Because I mean, we gotta mention this opportunity for students. You told me about that during summertime they have this neuroscience course, not just neuroscience, a lot of other options.
Yes.
But you specifically teach neuroscience.
Yes.
And you're gonna probably be there next summer teaching them.
Yes. So I do teach. Somehow it ended up happening over the years. I don't know what it is. Maybe it's a bit of I can control things now in a way that I can do things the way I want.
I can manipulate the flights. I can manipulate the classes.
Yes, yes. And importantly, not manipulate the students, which I just feel like is what teachers do too often. That is not always true. I want to be very clear, but it's easy. It's hard to make a classroom achieve all the goals you want. It's really hard. And there's all sorts of like, oh my God, I'll get into what I actually do in a second. Just, I can't not go on this tangent. Like, it's just like, there's so many like programs of like, this is how you should teach science, or this is how you should like TA, you know, be a teacher's assistant, this is how you should do this and that. And like, the structure about it is just so bad, and it's so like strange, and some of it's good, but like, you know, very often I think people have professors that teach a class and they're like, especially if it's not their subject matter of choice, where they're like, oh my God, you know, that professor's just so smart. He couldn't even explain it to me. He just has so many things in his head. He's so smart that he couldn't even, no, that guy's just an asshole and like didn't put the time in to give a shit about how to explain the concepts to students in a way that's meaningful so they actually learn. And it's just
and they fall in love
such
with it.
a shame. Yeah, and like, you know, the feedback I usually get is he's so enthusiastic.
That's
all I get.
I was about to give you, like,
yeah.
When you start with the fly, with the courtship, I think I'm already imagining the short video about you showing the courtship.
For sure.
Fly.
That's a horrible thing to know, but you know, I got quite lost in it because,
Because
yeah.
you're passionate about your work.
I guess, yeah, or I'm just really weird. It's one of the two. But like, you know, but you could explain really important.
You gotta be a little bit weird in order to do all of this. You gotta have a little bit of
sure,
that.
without question, because it's an odd line. It's an odd line of work, and it's barely work, but certainly is labor, you know.
But let's talk about your spare time. Spare time. Do you have your own time? Because you're in your lab a lot.
Yes.
Yes. What comes with the rest of your life? The hobbies, the interests.
Yeah, I'm gonna give you the worst answer.
Your friend.
This is my hobby. This is my interest. Like, that's me though. That's not like, there's a bit of a, I'm going to use the word toxic. I hate using the word toxic. I also hate saying what I'm about to say.
Big word.
Yeah, I know. And like, people just like fall in line behind some particular narrative. But look, there is a type of narrative about I'm a PhD student and I'm going to work 90 hours a week and you're going to, you know, not. And like, that's what you should do. I think it's great for some people. I want to do that to myself. It's just the way I live my day, and it's just my interest right now. It's definitely not what you need to do. You do need to put a lot of time in. You can't just, I really think it's hard to work 40 hours. People find a way to do it. But anyway, I personally enjoy the way in which I spend my time, where I spend a lot of my time in lab. I like the people, I like the experiments, I like talking. A lot of my time is spent talking. More should be done doing experiments. Outside of that, I hang out with my friends. That's pretty much it. I cook.
And you go to Belgrade.
And I get to go to amazing places like Belgrade and experience people and eat just the best food in the world. It's just meat,
what are your
bread, and
impressions? Is it your first time, right?
First time.
And how did you like it?
I really loved it. It's a really impressive and special place. And I've barely seen it, you know, I've only talked to a few
Rammstein
people.
is here today, so you can.
all 100% of Rammstein's fan base from the entire world, and especially Germany, is now here. And I'm like, what is going on? I was like, where is this concert? I will not be going. I have other things to see. But I
visit us again?
I will, of course. I have to. Let me just finish talking about my class thing that I was totally supposed to say.
Okay.
Just because it's a useful opportunity, I think, for
the
people.
neuroscience thing?
Yeah.
Okay,
so just like it's a pre and this isn't an advertisement. They get enough people anyway. But
yes,
if
but
you
it's
can
a good
afford it,
advertisement for the kids.
if you can afford it, and there are scholarship programs, which there should be probably far more of. There is a like pre-college class at Harvard, a lot of universities and a lot of places in the U.S. and probably elsewhere, where you can take a course over the course of 2 weeks. That's like something you would take if you were in college, sort of like a small version of it. Learn something new. For example, my class is on, and there's a couple people, me and my friend also do this, is a class on kind of introduction to neuroscience through the lens of motivated behaviors. These things I've been talking about a lot. Obviously, it's the things I think about, and I wouldn't teach it if I didn't think I would have something to say. And it's 2 weeks. You get to stay in like the dorms, and again, a lot of universities have this.
Here's the Harvard life.
And experience, yeah, it's like not
and
like
put it in
like,
the CV.
It's important. And put it in your CV. And like, listen, it's not like the best thing out there, like, but it is. It is a, you know, you get to, as far as material is concerned, and just like having a nice experience, you do get to learn about things that you wouldn't normally learn about, and you get to meet some people, and it's a good thing. It's not the best thing. It's not everyone should be doing it. Certainly not everyone can afford it, but like, again, apply to these things, apply to these like scholarship programs. And, you know, it's not, it's one of many opportunities, maybe not the best, maybe not the worst, one of many opportunities that you can get to learn something more complicated and learn about a world that you wouldn't necessarily know at a younger age. And so again, it's like a totally like a bit of a privileged thing, but
and when is the application for that usually? Spring?
It's a little bit too late. Yeah, it's usually in the spring.
So for the next year, right?
For next year. They do it every summer, and it's not just my class. There's like so many classes that they do this.
And where can they find that?
If you just type in the name of a university and then type in pre-college program, you can see if they have one. There's tons of them, not just Harvard.
And it's open to international students also?
Oh, for sure.
Do you get a lot of internationals on that course?
Yes. Yeah. Yes, and I did something like this when I was the first neuroscience class I took. I did something like that in my time in high school.
It's good to know because, you know, most of the countries, for example, Serbia doesn't have like a neuroscience thing on the batch. You know, most of the countries don't.
Yeah, even a lot of universities don't.
We talked about it master's level. So you got to finish either psychology or medicine or something similar, or biology,
right.
and then you can
in which
progress.
You'll learn some things, but you know.
And then you progress to neuroscience. But if you tell the kids that there is such an opportunity where they can actually, you know, get to know what neuroscience is, and by a Harvard PhD student, but maybe future professor.
They technically call me professor, but I like
How do you feel?
Throw up in the back of my mouth, and I'm like, please call me by my first name. Please just call me Ethan. It's like sort of like a knee-jerk reaction.
When you talk to them, does it remind you when you were young?
No, these kids are far more motivated than I was. These students are like so much more like sitting there. They all ask questions. I was just like, I mean,
why
I did
am
it
I
too.
here?
No, I did it too. I signed up for it mostly for like fun and because my parents thought it would be a good thing to do. But no, listen. The classroom is like can be a sacred place, and it's just like done so poorly so often. And so I try to make it a good environment, and I try not to like be super preachy. Although boy, do I just go on all the rants that I continue to go on here. But yeah, I think it's.
But
a nice
this is your work, and you should be passionate about it. And you should share it. It is the opportunity to share it. Well, for now, Serbia, maybe the world. I don't know. You never know who's going to watch this.
For sure.
Yes. So
yeah.
I'd say sharing with the world, and you should do more of this. I encourage you. I mean, this is just the first. It's like a pit stop. And then now, let's see with maybe Andy Huberman for a bigger exposure.
I don't need that.
No?
Do I need that? I don't know.
Well, I think your work needs to be heard. And I think, you know, scientists, you know, often go in their labs and hide those amazing research that you're doing. And then we get, you know, different perceptions of what you're actually doing.
Yeah, well, that's an important. I mean,
because you don't get out much and speak,
and
we need you to go out and tell us, okay, we're here now, we're doing this, and this is probably the next step. We don't know much. That's the thing. I'm gonna say we don't know much. We're gonna know probably in a couple of years, but that's it.
I'm gonna agree with that message. Actually, I'm like pretty like against like a self promotion in like a lot of ways.
Don't look at it as a self promotion.
Right.
Look at it as a promotion of science.
Yes, right. And here's the problem to address, which is that science does end up being pretty like
commercialized,
sequestered. Well, that's a whole other thing. Sequestered in these big academic journals that you need to pay money to access unless you are part of an institution. And that is a whole debate that is going on right now, is how do you disseminate information as a scientist to like the public? Does the public always need to know? Probably. They're not always going to care. At least the big stuff. The big stuff eventually makes it to your life because it touches your life in some way.
Like corona.
Which I'm not going to talk about. In cancer research, obviously that it filters down somehow, and eventually you learn it as if something is so ingrained, the next generation learns it as if it's canon, and that's how everything accumulates over time. But it is a difficult thing.
We didn't have podcasts and we didn't have independent media before, so now is the time where you can do
It's a good time, and I would like to be the one, or I would like more scientists to be the one that does it, to talk about the things that they know about.
Yes.
And not just be an influencer and be like, I'm a Harvard PhD. Here's like what you need to know about, and then just start talking about random stuff that they read from some other article and try to disseminate information poorly. I think it's important to we should say
stick to what you know.
We should say what we know. We shouldn't just be out there just communicating about everything about science, because just like everyone else, we're gonna have some opinion.
Yes.
And we think we know stuff because we spend a lot of time doing it, but we're gonna be wrong about a lot of random things about the world and our knowledge that we can't be sure of. And my rant is...
No, it was amazing, and thank you for being so humble and down to earth, because kids and people watching this probably are going to expect just what you said, somebody from Harvard PhD talking about how awesome Harvard is, and being in Harvard is awesome. You shed a different light on this topic.
And I want to say my final piece on that, which is this is going to start off really unfortunate for a second, which is that, you have friends in jobs, and pretty much every single job in the world, you're like, that guy's really bad at his job. That guy's an idiot. How does he have that job? That exists at every level, and every scientists are like that as well, where someone makes it in a career or has a position, but they're really bad at it. And I'm not saying that to put people down, but the point is that we are fallible. And including at Harvard, you're not just there and it's like magic happens. It's like, there are good people there. There are good people there and there are bad people there. And there are really good, really, really good people doing really the most important work. Obviously not just at Harvard. I can't say that enough. I'm even sounding like I'm being too acknowledging about that, to the point where I'm losing my point. It just exists everywhere. And there's a privilege to being in a place like that. But there are people who are really bad scientists who are there as well, and no one I know personally, but it's a thing.
Everywhere. Yeah,
it happens everywhere, and it is a privilege to be there for obvious reasons. But this is, I think, an important nuanced point for everyone to know.
And thank you for sharing.
Yeah, hopefully that's not just awful and insulting.
No, it's not awful and insulting. Actually, it's so refreshing. I would say refreshing is the word. We don't get perceptions like that.
Yeah.
Usually, people are snug about that, and they're like,
Yeah, and
snobby.
again, it is a privilege. I gotta say, I didn't think I'd end up there.
It is, and we know it.
But it's a silly one.
But people over glorify it.
Yeah.
In terms of it's kind of that people forget that also you're humans. And
for sure. Sometimes they forget it too, to be totally honest.
Can I touch you?
Yeah, you could hit me.
Your
I don't care.
Professor Dragana told me I have these students when I enter the room, they're standing up. They're like, why are you doing that? What's wrong with you? Don't stand up. Chinese students or whatever, she was telling me the story. And it's like, please, don't do that. It's like, I'm just a human like you.
Right. No, it really is. I mean, there's an authority complex that is different from different places that you're from, and that professors have.
Tell me about it in Serbia.
Yeah, and medical doctors are like, you got to call me doctor. And a lot of scientists are like, call me by my first name, man. That's kind of weird. That's me. Sometimes to the point of being overly annoying about it. But anyway. I said enough.
Ethan.
Yes.
I wish you luck in your research.
Much.
I hope I can get to visit that laboratory in Boston. I mean, I've talked with Dragana, I've seen you, I've met the team, and now it's time for me to fly there. Yeah, I need to. It's time. Now it's time to film the flies.
It's time.
I think it's.
I can send you videos if you want. I don't know what you're gonna do with it, but you want a clip? I'll send you weird clips of flies doing weird things if you want. Yeah. Absolutely.
Thank you. Yes, we
Thank
need
you so
it.
much.
Thank you so much for being here.
I appreciate it.
I appreciate you being here.
Thank you.
Dragi gledalci, hvala vam što ste ostali sa nama, nadam se do kraja epizode. Biće titlovi i pokušat ću da stavim timestamp da možete da vidite o čemu smo kad pričali. Probat ću da dovedem još ovakvih ljudi iz inostranstva, što mislim da nam je značajno, pogotovo za naučnu zajednicu i za studente, tako da mi se vidimo u nekoj od naših sledećih naučnih epizoda, a do tada ne zaboravite da se subscribe-ujete.